Yeah, well, let's go ahead and get started then. So, I'd like to thank everyone attending today. So, this is our kind of inaugural session that's open to the outside for the Center for Firefighter Health Collaborative Research. And that's at the University of Arizona. And I'm proud to introduce my colleague, Dr. Melissa Furlong, who's an environmental epidemiologist in our Environmental Health Sciences program. And she is uh, doing amazing things with the omics, which you'll hear about today research as well as firefighter cancer, reproductive, and other risks. So with that, I think you're all in for a treat. And what we'll probably do is keep the questions right, put them in the chat. And Michelle Valenti will be looking at that, and I think will help address those questions at the end of the presentation. Unless, Melissa, I figured with this size of a group, you didn't want interruptions during your presentation. Is that correct?
I think, yeah, generally we should hold questions, but if there's something burning about a specific slide, then it's okay.
All right. Well, thank you. All right. Off to you, Melissa. Thank you.
All right, thank you, Jeff. Okay, so what I'm trying to do today is make this presentation as relevant for firefighters as possible. Do you guys all see the correct screen? Yes, okay.
Yes.
So this is not specifically about my research. I'm covering a lot of stuff that the FFCCS has done. I'm pulling in research from some other groups and some other cohort studies. But I wanted to make this basically as relevant for firefighters as possible, but also from a fairly scientific perspective. Okay, so today we're going to talk about what is PFAS. We'll go through some of what we know about firefighter exposure to PFAS. We'll talk about toxicity signatures and known health effects for PFAS. And then in this, I'll get into a little bit of what we've seen in the FFCCS. And then probably what most of you are most interested in is how to reduce or mitigate exposures. So from personal, both personally and at work. All right, so what are PFAS? I think most of us know this, but the PFAS chemicals have really nice oil and water resistant properties, which make them good for industrial use in lots of different commercial products, right? So it's in paint, it's in water resistant clothing, it's in our stain resistant furniture, stain resistant carpeting, it's in cosmetics, photography, some pesticides, lots of fast food packaging, nonstick cookware, personal care products, and some firefighting foams. The thing that characterizes PFAS are these long chains of carbon fluorine atoms with the different groups at the end. So we know that PFAS is higher in firefighters, a little bit higher in firefighters, according to some studies. So this was not the FFCCS. This was actually in a study called HEROES, which is a study of essential workers. And the way we can read this chart is if we start over here at PFH excess, this dotted line means that there is no effect. So there was no elevated, if the effect is on this dotted line, it means that there's no elevated level. If the dot is over here to the right, that means it's increased. And if you look down here at the x-axis, this is somewhere between 1.2 and 1.3. And I believe this, for this particular study, it was percentages. So this means that firefighters had about a 28% a higher level of PFH excess than the referent group, which in this case was other essential workers. And this is just the effect estimate for other first responders and other healthcare workers. These lines by the dot indicate a 95% confidence interval. So if the confidence interval doesn't cross this null value, it means it was statistically significant. So for these particular PFAS, we saw significantly elevated associations in firefighters for PFH excess, SMPFOS, NPFOS, and PFHPS. These technically touched or slightly crossed the null, so they weren't technically statistically significant, but they're still elevated and concerning. And there are thousands of PFAS. So these are just some of the ones that are commonly detected in blood. Um, okay, in another study, this was by Jackie Goodrich at Michigan, who's a co-lead on the FFCCS. She looked at the PFAS levels in firefighters in the FFCCS between 2016 and 2019, and we compared them to the levels in NHANES. And NHANES is a national study run by the CDC, where for the last 25 years or so, maybe even longer, they have randomly sampled people from the population of the United States and measured lots of different biomarkers of different chemicals. And so here, these are the PFAS ones from the NHANES national study from 2015 to 2016. And you can see that the levels in the firefighters were higher than in the general population, right? 2.5 compared to 1.22 for PFH excess. They were slightly higher for NPFOA, NPFOS, SMPFOS, PFDA, and PFUNDA. So we saw slightly higher levels for PFAS for these, which is consistent mostly with what we saw on the previous slide. This was a study in San Francisco by Jessica Trowbridge, so not in the FFC. And she saw that these were significantly different and higher in firefighters for PFH excess, PFNA, and PFUNDA. These are not necessarily consistent with what we saw in the FFCCS or in HEROES. However, we do see that PFH excess has this consistent signal for elevation across the three different studies. One thing to note is that PFAS levels are declining over time in the population. So in NHANES, when they look at values from 1999 compared to more recently 2017 through March 2020, so just before the pandemic, that over that approximately 20-year period, there were declines of about 87% in PFOS, 74% for PFOA, 52% for PFHXS, and 16% for PFNA. And just a visualization of that drop, right? We see a really strong drop for for PFOS. And less distinct drops for the other ones, but still a decline over time. So this matters because sometimes when we compare some of our firefighter levels to NHANES, we don't see a difference. So this data was provided by Judith Graber, who's also a co-lead on the FFCCS, in a CAP study at Rutgers, which I believe is mostly volunteers. And when they compared the PFAS levels in the CAPs versus NHANES, they actually saw either no difference or slightly lower values in the CAPS firefighters. And part of this is probably just because of a mismatch in the years that were collected, and also some sites. Right. But so the moral of the story is that for the PFAS exposure is that even though the PFAS exposures are higher in firefighters, they're not dramatically higher across the board for all PFAS. They're slightly higher, right? Between like 20, maybe and 50%, maybe 2x for some of them. Okay. All right. So the next question is, where is the PFAS in the firefighter blood coming from? Right. So if we can know how they're being exposed, then if we know the important routes of exposure, this can be really important for mitigation purposes. So some of what we know, right? We know that PFAS is in the gear. We don't necessarily know that it's coming from the gear, but we know that PFAS is in the gear. We know that PFAS is in some firefighting foams. We know that it's in materials that are in structures and that burn during structural fires. Exposed. So we have to study that link. All right. So this data comes from Heather Stapleton's lab, who's also a co-lead on the FFCCS from her North Carolina study. Here they compared PFAS concentrations in the dust in fire stations compared to dust from their homes. And the way we can read this is the white ones are the fire station levels, and the gray ones are the levels in the homes. And here, this P less than 0.0001 just means that this is, again, a statistically significant difference. So they're much higher for PFH excess, it's much higher in the fire station dust compared to the home. And then when you look at the rest of the values, you can see that they're actually significantly higher in fire station dust for PFOA, PFOS, and PFNA. However, for PFDA and PFBA, those were not detected in homes. And then for these two, these were not actually detected in the stations. Um, and then, sorry, something check that. Um, so, so we know it's higher in the fire station dust. And then there was a study of World Trade Center responders and PFAS levels in their serum, and they compared the World Trade Center responders who were mostly exposed to smoke against those who were mostly exposed to dust. And what they found was that for PFH excess, the responders that were mostly exposed to smoke actually had much higher levels of PFH excess. And they also found that that was true for PFOA. So at least in this particular World Trade Center response scenario, it does look like smoke. There was something about the smoke that might have been contributing to some of the PFAS exposure. Okay, and so then this question about PFAS in the gear ending up in firefighter blood. And I would say this is somewhat of a everybody wants to know the answer to this question, right? So we know that PFAS is in the gear, but does it actually end up to PFAS in the gear? So does it actually end up in the blood? So we know that there are some PFAS that are intentionally applied to turn out gear, right? It includes these FTOHs, FTACs, FTMACs, MEFBSE, and PFBS. Um, However, many of these are actually not measured in the blood. And part of this is because, at least part of it is because they are fairly rapidly excreted, and which just makes them not good biomarkers in blood. However, PFBS is routinely measured in blood. It's not necessarily one of the most commonly detected ones, but we can look to see if there's anything on PFBS in relation to firefighters specifically. Um, so Jessica Trowbridge did include PFBS in her study of firefighters, and I apologize that these estimates are so small. Again, the dark blue are the women firefighters, and the light blue are the office workers. And this was not a significant difference. It wasn't even really close to being significant. It does look like maybe it's like slightly higher in the firefighters, but it's not not really at any meaningful level. We also looked at it in Heroes for PFBS. Here, the firefighters actually had lower levels of PFBS than the other essential workers, because when it's on this side of the line, it means it's lower. So we don't really, so for PFBS, we don't really know if the gear is a significant source of PFBS. For the other 8 PFAS that are applied to gear, we really don't know if those are ending up. We don't know if those are ending up in the firefighter blood. So this, I would say, is an unresolved question. Okay, so what is PFAS doing to us anyway? Right, so there are a couple of national or international bodies that sort of do a really good job at reviewing available evidence and issuing guidelines and guidance about health effects. And so one of them is the National Academies. And they issued some guidance on PFAS exposure testing and clinical follow-up a few years ago. And they basically determined that individuals who have blood PFAS levels that are greater than 20 nanograms per milliliter, where that's the sum of 7 different PFAS, should be screened for thyroid disease, high cholesterol, and kidney and testicular cancer. So they basically looked at the literature. They said, okay, these people are going to be at higher risk for these outcomes, and they should be monitored. So that's one governing body. Another governing body is IARC, and that's the International Association for Research on Cancer. And they convene experts all the time, many times a year, to make determinations, review the literature on specific chemicals, and then classify those chemicals according to their carcinogenicity and basically determine if they are carcinogens or not. And not just chemicals, right? So IARC is also the group that made the determination about occupation as a firefighter being a carcinogen. So what they determined is that PFOA is a group 1 carcinogen. That means the most possible evidence for carcinogen. And PFOS is a group 2B possible carcinogen. And they determined that there was sufficient evidence for kidney cancer, suggestive evidence for testicular cancer, limited and inconsistent evidence for breast cancer. And so this is not like the full, complete world of cancers. It's just the cancers that had sufficient evidence across different studies to make it into the IARC monograph for this. So in terms of looking at other possible health effects of PFAS, there's a whole wide world out there of health effects that we have not necessarily looked at. And so I'm going to present some results from the FFCCS on what we've done to look at health effects of PFAS. And I think most of you are probably familiar with the FFCCS, but for those of you who aren't, it's a community-engaged prospective cohort study done with the fire service to advance firefighter cancer control and prevention, and to also look at prevention of other health conditions. The goal is to enroll 10,000 firefighters. Many of you have been really vital in our in achieving our goals. So our current enrollment is over 8,000. We couldn't have done it without all of the firefighter liaisons and all of the people working to make this happen. So we appreciate that. And then we collect biological samples at study enrollment every 2 years after specified exposures. And then we also do an annual follow-up survey that some people are better than others at filling out. Okay, so obviously, well, maybe not obviously, but the bulk of the firefighters have been enrolled just in the last few years. And so there's not enough time basically for those firefighters to have developed cancer, for there to be enough cancers in the FFCCS for us to be able to study them in a scientifically valid way. So one of the ways that we're looking at cancer and other health outcomes is we're using um, biomarkers of gene expression, essentially. And so I'm just going to tell you a little bit about gene expression, and I hope this part isn't too painful. But essentially, but what we have is I'm just going to give you a little background here. So there's DNA. I hope most of you are familiar with the concept of DNA. Every cell in your body has a copy of your DNA. You were born with it. For the most part, you keep it throughout your whole life. But the DNA is turned into different proteins, and the proteins are what makes the building blocks of your body, right? It's what makes your skin cell different from liver cells, right? But the interesting part about that, right, is that your skin cell and your liver cell have the exact same DNA, but they look very different. And that's because the genes are expressed differently in different parts of your body. And also, even within a human, over time, genetic expression is different, right? So, like when you're 50, your genetic expression is different from when you're 20. And at 50, you're a different person, right? You have different body aches, you have different health conditions. You look different at 50 than you do at 20. And much of this is due to the way that the DNA gets turned into the protein. And so DNA in its journey to the protein goes through this process where the DNA is transcripted. It's basically um, taken apart. That the relevant section is copied into this mRNA, and then it's translated into this protein. And so we look at two ways that this process is regulated. So one way is by a process called DNA methylation, where it binds to the DNA and it affects this particular step. And another way is through a process called is through a compound called microRNA, which can bind to the messenger RNA and it can affect this translation. And so these two are called epigenetic modifications, and they can change the way the proteins are expressed. So they can increase proteins sometimes, they can decrease the proteins, they can delete the protein. The protein might just never be produced. And then the other thing that we can know about is we know where the DNA methylation is happening. We know where the microRNA is happening. So there are marks along the genome. They can be mapped back to a specific gene, and we know a lot about what different genes do. So we can infer the function or the health outcome based on what gene is being affected from the microRNA or the DNA methylation. I hope that wasn't too painful. Um, okay, so the pattern of age. So I'm just going to walk you through an example of what I mean when I say epigenetic age. So let's say we have this guy, he's 25 according to the calendar, right? He was born 25 years ago. He's 25 years of age. His epigenetic age can vary, and we'll consider four different scenarios. So the pattern of epigenetic marks on his DNA might suggest he's 15. They might suggest he's 25. They might suggest he's 45, and they might suggest he's 70. So for most people, your epigenetic age is going to be pretty close to your calendar age. So for most people, they will fall in this bucket of like, 23 to 28. Their biological age will match. For s- for a fairly significant number of other people, their epigenetic age might be higher or might be lower, right? So here, if his biological age says he's 15, then his biological age is 10 years younger. Here, if it says he's 45, then his biological age is 20 years older. He has a 20-year age acceleration. Here would be a 45-year age acceleration. This would be really rare. I'm not sure that we see this that often. Um, So in this scenario, he would be epigenetically younger than his calendar age, and here he's older than his calendar age, and he might be at greater risk for some adverse health outcomes. Okay, so again, from Dr. Goodrich at Michigan. She looked at, this is actually from the same paper I shared earlier, but she looked at the relationships between PFAS and epigenetic age. And there's a lot of different ways that different groups have tried to measure epigenetic age. They're all a little bit different from each other, but here each column is a different epigenetic age clock. And then the rows are the PFAS. So she found that PFH excess was associated with increased epigenetic age across these clocks. Also NPFOA and SMPFOS. All three of these seem to be elevated in firefighters in some of our studies. And then when she looked at some of the functions of the specific epigenetic markers that were changed, she found that some of those epigenetic markers regulate or have been associated with kidney cancer, prostate cancer, lipid regulation, tumor microenvironment, DNA repair, thyroid disease, immune function, and cell death. Um, right, so kidney cancer, we saw that in in the IARC monograph. We saw lipid regulation and thyroid issues in the National Academy's guidance, right? So this is all sort of consistent with what we've seen. Prostate cancer might be a little bit new. Between PFAS and microRNA in the firefighters. And we saw associations between these PFAS, sorry, associations between these PFAS and these microRNA. And they downregulate, the PFAS were downregulating all of these different microRNA. And so for the top hit, we observed that the downregulation is actually observed in several different cancers. It's also that downregulation is also associated with chemoresistance in breast cancer. And so the PFAS was downregulating it, which is consistent with possibly an increased cancer risk. And then for the LET7 family, which was several of these were different members of the LET7 family, they're also downregulated in several cancers and downregulated with PFAS. And then we did a pathway analysis where we looked to see if the pattern of microRNAs were indicative of risks for other diseases. And there were a lot. This figure was unfortunately very large in the original paper, but you can see SMPFOS, PFHXS, PFDA, and NPFOS were responsible for all of the pathways here. And some of these pathways were including Alzheimer's disease, some hormone signaling, neurological pathways, lots of different cancers, detoxification, actually, cytochrome P450, diabetes, and maybe some more thyroid cancers. So fairly consistent evidence PFAS, uh, for PFAS influencing genetic expression, consistent with what we've seen in the larger health effects world. All right, so just we also so I mentioned we were doing the epigenetics. I mentioned how DNA turns into proteins. So here we looked at the protein, the actual protein expression, so not the changes in the genes, but the changes in the proteins themselves. And this again is in HEROES, which was the cohort of essential workers. So this is not necessarily in this is not all in firefighters. I think some maybe like 20% of them are firefighters, but don't quote me on that. So we when we look when we these are just the results. So we saw associations for many of the PFAS with this group of proteins. So it's this hepcidin antimicrobial peptide, ferritin light chain, ferritin heavy chain, and another ferritin light chain protein. Um, so this was a little bit kind of interesting because I hadn't actually seen anything on PFAS and ferritin. Um, the associations are mostly driven by branch chain PFOS, and PFNA, which are not necessarily some of the ones that we're super concerned about in firefighters, but obviously are relevant for the general population. So when we looked into the ferritin light chain, this is not published yet. There was a paper that was published 2 weeks ago that actually suggested it was in mice, and they found that ferritin light chain is a key regulator of age-related cognitive impairment. So they found that when they increased expression of ferritin light chain, that they reduced the cognition. They also reduced some of the they also changed the way that the brain cells acted in a way that was consistent with aging. The good news about this was that when they reversed the FDL1 increase, so when they took it out or they removed it, they actually found improvements in age-related cognition and the brain markers. So it suggests that any damage uh, might not necessarily be permanent here. All right, so coming soon, that begs the question of whether PFAS is associated with cognitive function. We don't really know that. That is, well, we're starting to do some of the analyses, and we need to present that to the participants in the study before we share it more widely. Okay, so PFAS mitigation, the exciting part. Um, I will say that the following slides were contributed by two wonderful graduate students, Reagan Connor and James Hollister, who did the analysis and wrote up most of it. And Reagan actually pulled these slides together, and she presented them last month at the International Society for Environmental Epidemiology. So there is more data on PFAS mitigation out there in the literature. I'm just sharing what we're about to publish from the FNCCS. So this includes about 2,000 study participants across 7 US states, 30 fire service agencies, and they were on average 40 years of age when these samples were collected. And we we collected blood between August of 2023 and October 2024, so fairly recently. And we measured for 20 different PFAS analytes. And then we also had a baseline comprehensive survey of exposure. So we asked them about possible sources of PFAS, different occupational behaviors that might be associated with PFAS, we at the individual level. We also did a fire agency survey. So we asked the liaisons within each agency to complete the survey of agency policies and resources, and that was to sort of fill in the gaps for agency policies that the individual firefighters might not know about or they might be incorrect about. We did some statistics. You can ignore that mostly, but we had these PFAS analytes, so total PFOS, the branch chain, linear chain, total PFOA, LPFOA, PFH excess, PFNA, and PFHPS. Just in this analysis, in the paper, there's going to be more PFAS analytes included. And then we're going to share results of the non-occupational predictors, the occupational predictors from the individual survey, and then the occupational predictors from the agency level survey. Okay, let me orient you a little bit to these results. So On the left side, we have the category that we're looking at, right? So on this, we're doing non-occupational predictors. We have race, we have this history of donating blood or plasma, and we have home drinking water. And so for each category, we have the relative change in PFAS concentration for the different PFASs, right? So this first column is going to be total PFOS. This is going to be BR-PFOS, L-PFOS, total PFOA. A green means it's associated with a reduction, and the red means it's associated with an increase. Okay, so race here is actually sort of inconsistent so for blacks, they have higher levels of LPFOS, but lower levels of total PFOA or LPFOA. And that's relative, sorry, the referent group is here, white non-Hispanics. That's relative to white non-Hispanics. Hispanics seem to have somewhat lower levels of PFAS, and then this other group may be slightly higher. So then we see that for history of donating blood or plasma. So this question is just have you or do you donate, have you donated blood or plasma? And we see fairly consistent associations with reduced PFAS across the board for all the different PFASs for history of donating plasma. And then for blood and plasma, we see also decreases, but they're not quite as dramatic. For home drinking water, we saw decreases for bottled water, but increases for well water, which is consistent with what a lot of other studies have seen, and maybe some decreases for other. But I think this was just other types of water filtration systems. All right, so that's non-occupational predictors. At the individual level, so this is what people are reporting that they do. So we saw increases for career firefighter years. So the longer you're a firefighter, the slightly higher your PFAS is. Increases for military service, history of military service, current AFFF use, which is also consistent with what other studies have seen. And then for on-scene decon, we see these decreases for soap, rinse, brush, and rinse only, compared to no on-scene decon. And then maybe some differences by rank too. So slightly higher levels for captains and driver operators. When we looked at the agency level occupational predictors, we saw some interesting effects. So here, the policy for washing gear prior to storage is associated with a decrease in PFOA. A gear bag is associated with a decrease. Carpet is actually associated with an increase. And this is also sort of consistent with, I think, maybe some other PFAS that Heather had reported in a previous study. Reverse osmosis filters were associated with decreases, and consistent station dusting was also associated with decreases. So I know that's a lot to remember. So Reagan put this very handy summary together, where we observed lower PFAS associated with history of blood or plasma donation, on-scene decontamination, drinking bottled water or reverse osmosis, gear bags for all employees, this policy of washing turnout gear prior to storage, weekly station dusting, which I think most people would not be happy necessarily about, but yeah, weekly station dusting associated with reduced PFAS. And then we observed that higher PFAS is associated with rank, carpet in stations, drinking well water at home, AFFF's use, and military service. And we have a lot more variables that will be in the paper, but I didn't want to include them here just for to be a little more concise. Okay, so future findings to look forward to are we have this randomized controlled trial of plasma and blood donation on PFAS. The last participants should be finishing up in December, so we hope to be able to share those results from the trial in early 2026. And we'll also share findings on the plasma and the blood donation themselves and the health outcomes and the PFAS reduction. And then, many other studies to look forward to. I'd like to just acknowledge everybody who has contributed, which is just a vast universe of people across the country who've been very helpful and instrumental in making all these findings data happen. Okay. I ignored most of the chat questions, but let's see what we got. Michelle, are you moderating the
Yeah,
chat
and Crystal, so
question?
I
Okay.
can go through. So in the chat, thank you to Crystal too for looks like you've addressed some of the questions. So one of The questions was on the Rutgers studies you presented from CAPS, and the differences, perhaps there could be differences between volunteer and career firefighters with those results. I think, Crystal, looks like you addressed that question. And then there was a question on the exposure in
Wait, wait,
dust.
wait. Sorry. Is Judith here?
No, I did not see her on the call.
Is anybody from Rutgers here? It is volunteers. Yeah. So that could also be a difference because that could definitely also be a difference. But usually the volunteers have slightly higher exposures. Actually, don't quote me on that.
Melissa, that may be that. This is Ben Waller. That may be backwards because, especially if their sampling was mostly from New Jersey, they have a lot of volunteers there, but they tend to have much more fire load in much older buildings in the city. So there's the potential for that variable to go either way. I would not, without studying, I wouldn't try to speculate
Yeah.
a whole lot.
All right. Yeah. I think Judith had mostly speculated in her email that it was a year situation, that it was just a year mismatch between the and Haynes data. Yes, Jeff?
Yeah, I was just going to say too that as Melissa showed, there was a significant relationship with using a reduction with drinking bottled water, which suggests again contamination of the drinking water. And I don't want to replay an old stereotype, but I've heard there is contamination of some of the groundwater in New Jersey, right? So I think that could be a big part of it too, even separate from the firefighting.
Well, New Jersey is known as the hazmat state, right?
Exactly.
Sorry, Michelle. Keep
Oh,
going.
no, thank you for addressing that. And then the next question we have is from Derek Beering on the exposure from dust in the stations. Where is this dust coming from? Is it the ventilation system, building construction items containing PFAS, being distrusted with daily fire station living? There was a comment that perhaps the PFAS potential source is coming from fireground. It looks like Crystal shared Dr. Stapleton's paper that discussed on it being like the building itself, newer versus older buildings, larger, smaller buildings, and carpeting as factors. Are there any other factors for PFAS exposure in the dust that you think are important to consider of where those exposures are coming from?
Yeah, I think that's a really good question. And it's one that we haven't really answered yet. But Heather, if you want to provide some more information, you're welcome to. Is she here
Yeah,
still?
I'm here, Melissa. Can you hear me okay?
Yeah.
Okay. Yeah, hi everybody. Yeah, the figures that Melissa was showing were from a study that we conducted at Duke University in collaboration with the Center for Environmental Health in California. So some of those samples were collected in fire stations in Canada, and some of those were from fire stations in different areas of the US, many in California specifically, with vacuum cleaner bags. We didn't have a good full detailed amount of information on the fire stations themselves. We had some information on like where the gear was stored, whether the rooms were carpeted or not, the age of the building. The biggest predictors, and I think you had it on here, Melissa, were things like whether there was carpeting or not, and a suggestion about that maybe how the gear was stored could be influencing levels as well. But it's quite possible that the PFAS is coming from building materials in the fire station. It could be coming from the carpet itself. Some carpets are treated with PFAS. And then there's the question of what's maybe brought in from the fire scene. There's a lot of variability and variables that could explain that we can't necessarily parse out one by one, at least with the data we have right now. I hope that helps.
Thank you, Heather. It was much better than I could have.
And then going through for everyone, I am recording this. If you would like a copy, please you could email I'll put the email in the chat. And then I'm not sure, I would assume we'll be able to post this to our website too, as well. Our next question is from Bowman, who's going to start donating plasma. They read about the firefighters from Australia that donated every 6 weeks. What blood test should they request for their initial level? Also, what procedure would you recommend for donation? Every 6 weeks or how often?
So the Australian study is sort of sparked the impetus for our RCT, our randomized trial that we're doing right now. In Australia, they did a plasma donation trial. They also did blood donation, and they measured their PFAS levels at enrollment, and then they measured after a year of regular donation. And so they in Australia they did it, yeah, they did it every 6 weeks. They donated plasma every 6 weeks. In the United States, if you're going to donate plasma, um, they allow you to donate, I believe, up to 2 times a week. It does. So the way the PFAS binds to albumin and globulin, which are proteins in your blood, and so when you remove those proteins, um, your body has to regenerate the proteins, and then the regenerated proteins theoretically will scavenge some PFAS. So there might be a time period where you have to wait for your body to regenerate, um, for your body to regenerate the proteins and to also bind to some of the PFAS in your body. So um, and it can take a few days for your body to regenerate most of those proteins. So there is some frequency, right? It's probably more frequent than 6 weeks, but it's probably greater than once a week, at which the frequency is most effective. They studied every 6 weeks. In our current trial, we're studying every 6 weeks. I think that's safe. If you need to do it every 4 weeks, just to remember, like the first of the month, whatever, that's probably just fine. Um, I wanted to say something else about the PFAS donation, but I can't remember. Oh, as far as testing your own personal PFAS, I've gotten questions about this, and I don't have any companies that I can recommend for personal PFAS testing, and I'm not even sure that that's available. Jeff, do you know?
Yeah. Oh, double clicking here. Yeah. So, Eurofins has their home test kit. I believe that that's commercially marketed and available to firefighters. And, it uses a little capillary tube. You prick yourself and you collect it and send it in. And they measure a lot of different PFAS chemicals. So you could use that if you wanted to. And my understanding too is that there are other commercial providers and labs, and I think that will get more common over time. But if you can't find the Eurofins or another location, again, feel free to email us at COPH, hyphen FFCCS, and we're happy to help answer your question.
Okay, and I remember what I was going to say about the plasma donation. So a lot of people are concerned that when they donate plasma, that their PFAS is just gonna end up in the recipient of the donated plasma. If you go to a company like CSL or BioLife or something, where your plasma actually ends up being processed for use in a medical product, and that plasma is filtrated and screened at multiple different levels, and they remove everything that's attached to it because it's not just PFAS that attaches to the proteins in plasma. It's like medications, all kinds of other toxins like viruses, bacteria. So they have to get rid of all of that. They purify all of that. I don't think that that's true necessarily for the Red Cross, but those people are getting like life-saving plasma treatments. The companies are filtering and purifying the plasma before they sell that. Okay, for this question about working out in turnout gear, it looks like Ben answered that. However, the IAFC recommendations are actually to only wear turnout gear when you're responding to fires. So you can use the IFF recommendations for not working out in turnout gear. There's also other stuff on the gear that's not just PFAS. Oh, and Heather put a link to the Eurofins testing kit in the chat. Okay, are there other questions? All right, maybe we can end a little early.
Yeah, don't if you have any questions that you think of later, I did put our email in the chat, the coph-ffccs@arizona.edu. Please feel free to reach out with any questions that you have. And again, I am recording this. If you'd like the recording, please reach out and let us know.
Okay, I actually have one last comment too on the working out and turnout gear. On the NFPA also has recommendations for non-structural firefighting PPE for firefighters for things like extrication, structural collapse, other forms of technical rescue. And those are primarily designed to protect you from cuts and bruises, bloodborne pathogens, exposure, and the weather, and not necessarily structural fire or flammable liquids type heat like firefighting turnout gear is. So that's another way to reduce exposure to PFAS from wearing structural firefighting gear.
Jean, I am not going to APHA. Is anybody else? Okay, are we going to share these slides?
I mean, I think we can share the presentation, and if you're comfortable sharing the slides, absolutely those as well. I mean, it's totally
Okay.
up to you, Melissa.
Yeah, those slides were adapted. Like some of those slides were from Heather, some of them were from Reagan, so we should probably ask them
Okay.
if they are okay with it.
Well, I just, if there are no more questions for Melissa, I just wanted to thank her. We'll see if there are any others here. Um, Yeah, I really wanted to thank her for taking on this task of synthesizing the information about PFAS. If again, if folks have additional questions, let us know. Um, and we're hoping to have these type of sessions on various topics from research, a lot of it coming out of the firefighter cancer cohort study at intervals throughout the year. So maybe, twice a year, maybe 3 times a year. So please look forward to more. And we again appreciate everyone joining us. If you have suggestions and comments about how to make this even more useful, please let us know as well.
Thanks, everybody.